All right, welcome to the November 2020 webinar. My name is CK Eternity, and this is the first of what I hope will be many of these webinars in the future. I plan on doing these about once a month. Somewhere in the middle of the month I'll be releasing each one, and maybe add some shorter videos in between or other things in the future. We'll see. This one is going to be covering the balance of iron and copper in living organisms, specifically the human body. I'm going to be unpacking a lot of what I've already talked about on Twitter and my website, but I'm going to be going into this in a little bit more depth and bringing up some things that I haven't discussed so far. So I hope you guys enjoy this, and without further ado, let's jump right in. So the first thing I want to talk about here is what's called the Great Oxidation Event. This was actually the first major extinction event to occur on planet Earth, and it happened roughly 2.4 to 2 billion years ago. At this time, the Earth's population was entirely single-celled organisms, living in really a much more shallow version of the ocean as we know it today. So interestingly, this extinction event was driven by a single factor, the increase in oxygen in the environment. And this happened because there was a big increase in the population of photosynthetic bacteria a little bit before 2.4 billion years ago, and they put off more and more oxygen as a byproduct of photosynthesis. The problem with this is that the increase in oxygen changed the Earth's atmosphere from what's called a reducing atmosphere to an oxidizing atmosphere. For those of you that aren't familiar with a lot of chemistry terminology, basically when a material loses an electron, we say that it's been oxidized. If you put an electron back into that material, we say that it's been reduced. So oxygen acts as a fairly powerful electron acceptor. When the oxygen levels increase, it would pull electrons from molecules or atoms and was highly reactive with them. And cells at this time weren't used to the increased levels in oxygen, so they were really prone to molecular damage as a result of this. One of the biggest factors that actually caused this damage was iron. Now there are two types of iron in biology. There's the ferrous form, which is iron 2+, and then there's the ferric form, which is iron 3+. The ferrous form was the form that's more commonly used around this time, and the reason for that is because it's the form that's water soluble. The ferric form, on the other hand, is insoluble in water, but it's a little bit more oxidized and therefore more stable. The ferrous form is unfortunately highly reactive with oxygen. So this actually is what drove a lot of the die-off during the Great Oxidation Event. And that's because when ferrous iron reacts with oxygen, it forms what's called iron oxide. Chemically, this is essentially rust. So when iron oxide is formed, it catalyzes a series of chemical reactions known as the Fenton reaction. When this Fenton cycle starts, it results in the creation of free radicals, which are, for those of you that don't know, essentially just simple molecules with an unpaired electron. This makes them highly reactive and very oxidizing. A lot of them are based on oxygen. Those are known as the reactive oxygen species, but there are others based on nitrogen and other atoms as well. So when the Fenton cycle is catalyzed by this iron oxide, it's especially destructive because not only does it result in the creation of a few different free radicals, but it also ends with one of the most harmful free radicals for biology, and that's the hydroxyl radical, which is a hydrogen and an oxygen. So this free radical essentially will damage any molecular structure it comes in contact with, and it lasts a very short time, so it's very reactive. Antioxidants may not have the time to clear it before it damages a lipid membrane or DNA or a protein structure. So this can be extremely harmful, and it was actually enough at the time to kill off most of life on Earth. Now, one thing that interests me is what sets apart the bacteria that survived this Great Oxidation Event, this massive extinction. I think, interestingly, they did this using copper. And one of the best examples of this we see today actually went on to become the human body and mitochondria. So let me unpack this a little bit. The enzyme that was really pivotal here is an enzyme called cytochrome C oxidase. This is what I consider the most important enzyme in the human body, and it's present in mitochondria as well as really most life on Earth, bacteria, plants, fungi, everything. So what makes this enzyme so unique and important here? Cytochrome C oxidase has the ability to convert oxygen into water. And interestingly, this is how mitochondria actually harness oxygen and use it to metabolize calories from food into ATP. So this is really one of the most important reactions that the body carries out because first off, it allows it to manage oxygen so that it doesn't create harmful free radicals and basically tear your cells apart. And secondly, it's also hooked up to energy production. So cytochrome C oxidase basically works using copper, and it also contains iron proteins in the center, and it uses these to shuttle electrons through this protein, and at the same time, it pumps out protons into the inner membrane space. And this is how it basically sets up the production of ATP elsewhere in the mitochondria. So this is part of what's called the electron transport chain in mitochondria. I'll expand this in a future video, but really the electron transport chain, to put it simply, is the series of proteins that mitochondria use to carry electrons across their membrane, pumping protons out, allowing them to later harness that proton energy into the production of ATP. So cytochrome C oxidase not only allows the management of oxygen, but it's also the first major example I can find where we see copper and iron working together. And the reason that these are so useful together is because they both have a really high potential to be used in redox reactions. So they both have the ability to gain and lose electrons fairly easily. Copper, being very conductive, seems to be a little bit faster at this. So it's often used to manage iron, which can be very difficult to reverse the oxidation on, to reduce it back down to the ferrous form from the ferric form. So let me expand on that a little bit. So there are a number of other enzymes that also use copper in the human body. Two of the oldest ones are actually antioxidant enzymes, and those are catalase and superoxide dismutase. Originally, cells used more simple structured antioxidants, such as melatonin and vitamin C, but as they developed and evolved over time, they created some more complex antioxidants that were highly targeted towards some of the specific free radicals that were formed within that cell. So catalase is one that's highly selective for the free radical hydrogen peroxide, and superoxide dismutase is highly selective for another called superoxide. Hephaestin is another copper-dependent protein. This one is a transport protein that is actually what allows iron to be carried from the enterocytes into the circulatory system. This is part of how iron is absorbed through your digestive tract and put into your bloodstream to be transported wherever it needs to be. The reason that copper is required in this case is because for this to occur, iron has to be oxidized down from the ferrous form into the ferric form so that it can bind to the specific transport proteins. Copper is also used in a few different enzymes within the nervous system in the brain. The most notable is probably monoamine oxidase, which is actually the enzyme that breaks down all of the monoamine neurotransmitters. You may have heard of most of these. These are dopamine, serotonin, melatonin, noradrenaline. Without this enzyme, these would really persist in the brain and not really be broken down at all. The other enzyme that copper is used in the brain is dopamine beta hydroxylase. This is the enzyme that metabolizes dopamine into noradrenaline, which is also really pivotal. Noradrenaline is an analog of adrenaline that's another one of the primary neurotransmitters in the brain. Another enzyme called tyrosinase uses copper. It's what converts tyrosine into melanin, so it's what allows tanning in the skin in response to UV light. It has some other functions as well, stimulating some of the different copper-dependent processes in the body, but we won't really get into that here. Lastly, probably the most important copper-dependent enzyme in the body is what's called ceruloplasmin. Ceruloplasmin is probably the best example of a copper-dependent enzyme regulating iron. Ceruloplasmin is also where we see most of the copper in blood plasma stored, actually. It's estimated that about 95% of copper in the blood is in the form of ceruloplasmin. And it actually is a protein chain. It's a fairly large protein that carries 8 copper atoms, and it uses them as some of the primary cofactors for the conversion of ferrous iron into the ferric form of iron. So the reason that it does this is actually so that that iron in the ferrous form is able to bind to the transport protein transferrin, which allows it to be transported really anywhere in the body for any purpose necessary. So without ceruloplasmin, iron metabolism is nearly impossible. Ceruloplasmin also has been theorized to have some other functions. It may act as an antioxidant in some situations as well, and really it's still being studied. We don't know the full extent of its activities. It uses either sulfur groups or oxygen as a cofactor, as well as the embedded copper atoms in the structure. So I've been talking quite a bit about some of the copper-dependent enzymes. You're probably wondering, what about the iron side of things? Interestingly, iron is actually the most abundant mineral on planet Earth. Most of that iron is going to be in the Earth's core, but there's a large amount of it that's also present on the surface. And because of this, it's been one of the most fundamental nutrients really since life began. In the human body, there's a number of different iron-dependent proteins and enzymes. Probably the most well-known is hemoglobin. This is a protein that utilizes the tendency of ferrous iron to bind to oxygen, actually for the benefit of your body. Hemoglobin acts as an oxygen transporter. Ferrous iron, there are 4 ferrous iron atoms present in its center, and these bind to oxygen and carry it throughout your body, allowing cells to use it to produce energy, which is very, very beneficial. Most of the iron in your body is actually present in hemoglobin, a little bit over 70% if I remember correctly. The protein that allows iron to be transported around, this one isn't one that's dependent on iron per se, but it is one that is very pivotal in iron metabolism, and that is transferrin. Transferrin is the main protein that carries iron to different parts of the body. So, for example, if a blood cell starts to break down or is damaged in some way, transferrin steps in, picks up the iron excess so that it doesn't end up as free iron and becomes damaging. It's picked up by transferrin and carried to the marrow of your bones or other parts of your body for either storage or the production of new red blood cells and heme proteins. Ferritin is the storage form of iron. This is where iron is deposited in the tissues. Something like 1/4 of the iron in your body is going to be put in ferritin at any given time. You have about enough iron stored in ferritin to last you, depending on your gender and your age, between 6 months and even a couple years in some cases. Hemosiderin is another kind of interesting iron storage protein. This one is going to be basically insoluble and inaccessible. So you can think of this almost as an emergency store of iron. This is where when you have a state of inflammation or oxidative stress or some other form of damage, that iron that would normally be oxidative is going to be shuttled into hemosiderin, where it's basically locked up, useless to your body, but still out of the way so that it doesn't cause more harm. Hemosiderin may play a role in a couple different diseases, but we really don't have a lot of data on it, mainly because it's very difficult to measure your levels of this. You essentially need a tissue biopsy, almost like a surgery, to be able to measure levels in any given tissue, and it's either very painful or very difficult to carry out. So hopefully in the future we have a little bit more data on that one, but it's not something that's well understood at this time. Cytochromes are some of the most common proteins you might hear about in the body. Really, all cytochrome enzymes or proteins are going to be iron dependent. These are primarily enzymes used to shuttle electrons. So they use the heme iron clusters to carry electrons and facilitate different reactions. We already talked about cytochrome C oxidase. This is one of the best examples of this. Ferroportin is another not really iron dependent protein, but something that really plays a big role in iron metabolism. This is actually an entry channel that's used to carry iron through cell membranes, mainly through the enterocytes, where iron is absorbed through the digestive tract and put back out into circulation, and also carrying it out of circulation into cells where it can be stored or utilized. So ferroportin basically is a doorway for iron, and generally it's paired with hephaestin, that copper-dependent enzyme that allows it to be converted back and forth between the ferrous and the ferric form so that it can be attached to transferrin or used for different purposes. The last kind of class of enzymes that are heavily iron dependent or iron related are the ferrooxidase enzymes. These include ceruloplasmin, hephaestin, and ferritin. These are pretty much any enzyme that converts iron from its ferrous form into its ferric form. So these are enzymes that, as the name kind of suggests, oxidizes iron. And these are really pivotal. Really, any enzyme that's carrying iron is considered the active form or the ferrooxidase form. So ceruloplasmin acts as a ferrooxidase when it's being used. Same with hephaestin, same with ferritin. So let's take a moment to step back and look at the big picture. How do iron and copper fit together? What does this metabolism look like on a larger scale? When you ingest iron, it goes through your intestinal wall into what are called epithelial cells or enterocytes. From there, the iron is pretty much going to be used directly by those cells, stored as ferritin, or it's going to be put out into the bloodstream, the circulatory system. To be put out into the circulatory system, it uses a combination of ferroportin and hephaestin. That allows it to be converted into the ferric form, where it's loaded into transferrin, and from there it can be carried wherever it's needed. Transferrin also is used in macrophages. These are well known for being immune cells, but they also have a very pivotal role in picking up free iron, iron from damaged blood cells, etc., and storing it or channeling it back into the production of new red blood cells. So this again uses transferrin. So when you have a red blood cell that becomes damaged, a macrophage picks it up, uses that iron from the red blood cell, it's broken down, and it's either used by the macrophage, stored, or it's sent out again through the combination of the ferroportin and, in this case, ceruloplasmin, which has a similar function to hephaestin, and it's transported out into transferrin, which is then attached to the transferrin receptors on what are called erythroid progenitor cells. The names here aren't important, but basically this facilitates the process called erythropoiesis in bone marrow. So the macrophages allow that iron from the red blood cells to be channeled into the production of new red blood cells through that process of erythropoiesis. And transferrin plays kind of the middleman there, carrying that iron from the macrophage into the bone marrow where it can be loaded into hemoglobin proteins once again. So where does copper fit in? Hephaestin carries iron from the gut wall cells into the bloodstream. Again, ceruloplasmin then steps in and is what allows iron to be converted back into the ferric form so that it can be loaded into transferrin when it's put into red blood cells in the first place in the bone marrow, or when macrophages pick up damaged red blood cells, and that iron needs to be recycled. So overall, this process uses primarily iron-dependent enzymes and proteins, carrying iron from place to place, loading it into red blood cells, and recycling it when red blood cells become damaged. Copper acts as the middleman in these situations. Copper basically converts iron from the ferrous to the ferric form and makes sure that it's protected. On the side here, you also have the antioxidant copper enzymes that, if you do have free iron, act as basically a protective barrier, preventing the oxidative free radicals, such as hydrogen peroxide or superoxide, that are formed from actually damaging the cell in any way. So copper protects the cell from excess free iron and helps facilitate its movement really throughout the body. So that's a basic overview of how copper and iron work together in the human body. There's a few other nutrients that I haven't really covered yet, however. The three big ones that I want to unpack here before I wrap this up are sulfur, retinol, and vitamin C. Sulfur is used with iron in what are called iron-sulfur clusters. These are basically clusters of iron and sulfur atoms bound together with amino acids that are actually basically used as a pool of electrons that can donate or capture electrons for redox reactions, similar to iron and copper proteins that we've kind of already been discussing so far. Iron-sulfur clusters were only found in mammal cells fairly recently, so for a while we didn't know that these were present in the human body, and as a result of that, we haven't had a ton of research done on them so far. But based on kind of the limited research that we do have, we know that they require plenty of bioavailable iron and sulfur, so all the other factors that we've already discussed have to be functioning properly. And they also utilize the amino acid cysteine, which is a sulfur-dependent amino acid that is really most well known for its function in things like glutathione and some of the other major antioxidants like metallothioneine. Interestingly, the formation of iron-sulfur clusters is very dependent on the level of oxidative stress. So if you have a lot of free iron and a lot of that Fenton reaction levels happening, a lot of free radical formation, it actually will result in reduced iron-sulfur cluster formation. And I think this can have a big impact on energy metabolism. The next really important nutrient here is retinol. For those of you that don't know, retinol is the name for the active form of vitamin A. This is the form of vitamin A that's really only found in animal products because it has to be converted from the carotenes that are found in green plants for the most part. So retinol, after copper and iron, is probably the third most important nutrient in the regulation of some of the processes that we've been talking about so far. One of its biggest functions is it works together with copper very closely and allows copper to actually be used in ceruloplasmin. So without retinol, ceruloplasmin can't be formed properly and function correctly. A lot of the time, if you get a blood test and you have low ceruloplasmin, it can indicate retinol deficiency. And because of this, there's actually a few old papers from more towards the beginning of the 20th century showing the reversal of anemia using retinol or high retinol foods like cod liver or cod liver oil. The reason for this is because the increase in ceruloplasmin allows the iron to be used by transferrin. The other reason is that transferrin itself actually uses retinol as a cofactor. So any iron transport in the body and the ability of iron to be converted from the ferrous to the ferric form for transport all require retinol. These are some of the most important reactions in the body, and this is why retinol is such an important nutrient and why I really talk about it a lot on Twitter and my website. It also has a few other functions that aren't really directly tied to iron metabolism, but do have some interplay with them. For example, retinol is a primary fat-soluble antioxidant. So if you have a high level of iron oxide or a lot of Fenton reaction happening, then retinol can actually interrupt that cascade and prevent the breakdown of lipids in the lipid membrane or elsewhere in the cell. That creates a chain reaction normally. So retinol can interrupt that chain and prevent further damage, essentially. Retinol also plays a really pivotal role in vision and photoreceptor function in general. It's oxidized into retinal, spelled with an AL on the end instead of an OL, and that form of retinol actually is bound to photoreceptor proteins, both in your skin and in your eye. So it's really one of the most crucial nutrients. Another one of the most important nutrients for iron metabolism is vitamin C. One of the biggest functions of vitamin C here is it actually increases what's called the bioavailability of iron. This means that it increases the amount of iron that can be absorbed from the food you eat. The iron in your diet generally comes in two forms. There's the heme form, which is iron bound to proteins already, that's found in red meat. And then there's the ferric form of iron, and in some cases the ferrous form of iron, the individual iron atoms that are found in primarily plant foods, but a little bit in meat as well. So it's well known that iron from meat is absorbed more easily. Iron from plant foods in some cases can't be absorbed at all if vitamin C deficiency is present. And the reason for this is because vitamin C acts as a cofactor for an enzyme called duodenyl cytochrome B. And this converts iron from its ferric form into its ferrous form so that it can travel through the metal ion transporter that's used to pull it into enterocytes, or the cells that line your gut wall. Vitamin C also acts as a cofactor for another one of the most important enzymes in the body called cytochrome B5 reductase. When hemoglobin is used up for a long period of time, it can become oxidized, which converts it into another form called methemoglobin. This is basically hemoglobin, except one or more of the iron cores present is in the ferric form instead of the ferrous form. This oxidized form can't bind to oxygen the same way the ferrous form can, so red blood cells that have a lot of methemoglobin basically become useless as oxygen transporters. This enzyme, cytochrome B5 reductase, is what converts that methemoglobin back into its active form by reducing that ferric iron back into its ferrous form. This reactivates hemoglobin and allows oxygen transport to continue in the body. Lastly, vitamin C is one of the oldest antioxidants. It actually prevents free radical damage from a number of different radicals in cells and mitochondria, including iron oxide itself. So it can very much protect cells against damage if there is any kind of issues with iron metabolism. So if you're trying to get your iron metabolism back on track or correct what appears to be iron deficiency of any kind, then you really need vitamin C. It's one of the most essential nutrients here. So at this point, you may be wondering, what can I do with all this information? The biggest thing I'll say is if you want to correct iron metabolism, you want to make sure you're getting all of its cofactors. This goes for really everyone, whether you've been told you're anemic or whether you're just trying to protect your health from future cellular damage or stress from free inflammatory iron. The biggest thing is you just want to make sure you're getting nutrient-dense foods like liver, seafood, eggs, other types of meat, and plant foods as well. I personally am a big proponent of an omnivore diet with a wide range of foods. If you want a more detailed guide to how to correct your iron metabolism, I have a protocol on my website, which is listed here, titled Free Iron as a Cause of Inflammation. I'd encourage you to read through that for more information. And in the future, I'll probably be putting out some more videos on this as well. There are some aspects of iron metabolism that I didn't cover. The biggest thing that I left out here was the B vitamins and how they fit into things. The biggest ones that play into iron metabolism are thiamine, B12, and folate, but these don't really regulate iron metabolism directly. They more so regulate things like the formation of new red blood cells, and when that process is faulty, it impacts iron metabolism from a distance. Other nutrients like thiamine and magnesium also regulate mitochondrial function, which has a big role in iron metabolism as well. So all of the nutrients in the body really impact mineral balance, and especially the balance between iron and copper. But the nutrients that I've listed here are, in my opinion, kind of the core of this. I'll be putting out more information in the future on things like different causes of anemia and how to resolve some of these issues that we've talked about. But for now, I'll leave this as a basic introduction. If you made it this far through the video, thank you guys for watching, and I'll be putting out another one of these next month and in future months. If you want more content and information in the meantime, check out my website or follow me on Twitter. That information is listed on the slide at the end. Thank you guys for watching, and enjoy the rest of your days.